Hello everyone, my name is Ashfaq Ahmed and I'm working in the field of bioinformatics and structural biology. Before this, we have covered two videos, part 1 and part 2 for exploratory protein data analysis or protein research. So this is actually the part 3, or in other terms this is the part 1, because in this video we are coming to discuss about some tutorials. In part first I have discussed that what kind of functions protein are performing, and in part 2 I have discussed those functions in terms of research, that what methodology you can apply to do some work in those functions. So today is the first video, or actually it is the real tutorial of protein structure modeling. So let's begin that what is a protein structure modeling. So the basic question is, what is protein structure modeling? Simply to predict or generate a three-dimensional structure for a protein. If you have any protein sequence, and if you want to generate a 3D structure, or if you want to generate a simple structure, but actually a simple structure is also called a 3D structure, so don't confuse yourself with this word. So if you want to generate a protein structure, we call it protein structure modeling. And why you need to model a structure? So I hope, or I assume that you should know that why you need to model a structure, but generally I should speak that to comment on function, for impact of mutations, for stability, for mobility and fluctuation, or for any other domain comparison, you will need a protein structure. And if it is not available, in other case, you will need this tutorial to predict a protein structure or to model a protein structure. So actually this slide is all about those 6 methods by which you can generate a protein structure. The first one is comparative modeling, or also called homology modeling. The second one is fold recognition. The third one is ab initio. The fourth one is homology template free. The fifth one is based on machine learning approaches, and the sixth one is hybrid methods. So in this video I will cover the tutorial for comparative modeling or homology modeling. For these other methods I will come one by one in other videos, so stay connected with us. So for this, the first one is comparative modeling or homology modeling. Let's discuss that what is comparative modeling, or what is homology modeling. So comparative modeling or homology modeling is actually to give you an idea in the most simple terms. Beyond this I cannot bring any further simplicity. So what you should know, based on comparative modeling or homology modeling, we actually generate structures, but from amino acid sequences you provide. It means if you have amino acid sequence, then by this method you will generate a structure. The amino acid sequence which you have will be called query, or will be called a subject. So remember, the query or a subject terms will be used for the sequence you have and you want to generate a structure for it. It will be called query or a subject. The structure is generated from copying the existing or other structure. The program will generate a structure for you and it will copy another structure. So we will call that structure a template. So these terms, the query or subject is actually your sequence, which you do not have a structure, and the template is actually the structure the program will copy it. So from the above these two statements, what conclusion we can drag? It means you need to provide amino acid sequence. Then the question arises that from where you can take the amino acid sequence. Maybe most of you don't know that from where I should take the amino acid sequence. So the answer is you can take it from a database, or you can have it from your sequencing project. On this slide, you may have certain question in your mind, so I have tried to answer those questions. The first question you may have, that on what basis this method will select the template. Template is actually the structure which is to be copied. So on what basis the program will select a template? Because there are many thousands and 10,000s and even not in million but in thousands there are structures. So why the program will copy some specific structure? On what basis? On what rules? So the rules will be homology, and that is why the program or the methodology name is also called homology modeling. But what is homology? If you don't know, then what you should know what is homology. So actually, statistically speaking, homology is actually the sharing of amino acid similarities between two sequences. So if you have two sequences of amino acid, or even more than two, but for your understanding I am mentioning two. If you have two sequences and if you compare them, and if it contains some sort of similarities or identities in the sequence, so the borderline is 25%. So if those similarities are greater than 25%, we will call it homologs or homologous to each other. And explaining this point, the similarity point from the biological perspective, it will be like this, that homologs share a common ancestor back in time. So it means the program will select a template structure for your sequence to build a model based on homology, and if they are homologous to each other, it means back in time these two sequences, the one which we are using structure, it has also a sequence. So these two sequences are actually homologs, and they share a common ancestor back in time. So before to further waste our time, let's move to the comparative modeling tutorial, that how it works. So from now on I will work from my screen, so you can follow me. So the first step is that from where you will take your sequence. So for example, you don't know if you have a sequence from your sequencing project, that's okay. If you don't have a sequence, there are many databases, but the one I want to mention for you, that is UniProt, P-R-O-T, uniprot.org. You need to open this database. Now you can read this, find your protein. So here you need to type the name of your protein. Most of the students, they are saying that I don't know the name of a protein, but I know the name of a gene. Most of the time the protein and the gene has the same name, so just try it. So right now I'm just trying to just give one name. Let's say I say E2F1. For example, I give this name UniProt and just click search. So when you search it, you will have E2F1 protein from various organism. You can see here from mouse, from rat, from human, from chick, from Drosophila melanogaster, and again from human, from mouse. So the don't waste, I'm not going to waste further your time here. So for example, this is your E2F1 is from human, so click here. Once you click it here, so the next thing is you click it here, the sequence. You can see the sequence tab. Click the sequence tab and just leave this open. Now open another server. I will teach you this on web server, because most of you may not be so good in manual modeling. Therefore I'm teaching you on server. If you are interested, drop me in your comments. I will also make one video for modeler to be used in manual settings. But in the current video I will teach you in the how to use the online server for homology modeling. So just write here Swiss model, and when you type Swiss model, you will have this kind of you will have this kind of website in front of you. Just click start modeling. So when you click start modeling, you will come here. So now the second step is that it will need your sequence. So this is our sequence. We have it open. We have opened it. So the sequence, you can see it here. It contains almost 430 plus amino acid. So just come here, copy sequence. So I have pasted my sequence here. So the one thing you need to be clear here, if you want a sequence, if you don't want the whole sequence, just say you want the half sequence, not the complete sequence. Wait. Okay, because sometime student came and they are asking that we don't want the complete sequence, we want half of it. So it doesn't matter, you can copy half of it. Okay, sometime a student came and asked me that I have a mutation, how to generate. So that is very simple. For example, your mutation is you have clicked this copy sequence, copy your sequence here. I'm copying my sequence here in the text document. Just open it and paste it. For example, you have a mutation, this R is changed to P, so make your change here. Now copy this sequence and paste it there. Okay, so once your sequence is here, you should see such kind of different colors. It means the sequence is accepted. Okay, now name your project. Let's say I'm naming it E2F1, and email if you want to see if you want to give an email, so just provide an email. Now we have talked about the template. The program will now search for the structure to be copied, and that are called template. So search for template, you need to click it and just wait for 2 to 3 minutes, depending upon your internet and the size of your sequence. So welcome back. I had paused my video because it took almost 3 to 4 minutes, so that's why. Okay, once that process is finished, you will have this kind of screen. Here you can see the name of your project, E2F1. Here you can see number of templates, 50. Okay, but the important thing is this one. You can see so many templates here. You see, most of the people, they just click here, build a model. That is wrong, and later on they came to me or other experts and they are complaining that we did the same process, but the structure is some different. The structure is different. So what actually happened to them? I want to teach you here, and you need to take care of it. You see this blue bar, blue. This is called query coverage or sequence coverage, because we called a query to a sequence. So the sequence coverage or query coverage. So this is called the query coverage. It means that, for example, if this option is selected, only this part of the structure will be copied. The program is also showing you the structure which will be copied. This is a template. Okay, so here now we had submitted almost 430 amino acid, or 430 plus amino acid. Okay, and we are getting this structure which only covers this portion of our sequence. So it means if we build a model using this template, so this portion, I would say these 200 amino acid which are missing, and these 100 amino acid at the end are missing, and the ultimate structure, you will be surprised that it is not your protein. Actually it is your protein, but the middle portion. If you select this one, it will be more less. I mean, you can see here only this part. So the first thing, you need to be here, but if you select it here, if you just pay your attention, here you can see some smaller name, like 4-digit name, 2AZE, 2AZE, 1CF7, 1CF7, 7PKN7. These all are PDB codes. It means these are real structures, but in this case it is telling you that AlphaFold database. So the program is actually telling you that if you want to structure your complete sequence, we will use the template from AlphaFold database. So the AlphaFold database, they do not always contain good structures, and in this case, for example, this is our case. In this case, if I model, my protein will also look like this, which is not the correct folding. It does not seems correct folded protein. So if we face such kind of scenario, then it means in this case the homology modeling is not working. And if the homology modeling is not working, then we will move to the other methods. I showed you in the start that there are 6 or 7 different methods. The first one among them is homology modeling. So remember, for every sequence the homology modeling will not work, because the homology modeling will only work if there is a template structure. If there is not a template structure in the database, the homology modeling will not work. Even if I want to model with a sufficient accuracy, I can click this and I can model only this part of my sequence, the blue part, and it will be like this. So here I am just to teach you how to build a model. So once you select your template, if it is here, and if you are satisfied from the template, just click build model. Upon clicking building model, it may take 1 or 2 minutes, and after that you have to download your model with some sufficient information. So you can see they have modeled your structure. It says that oligostate is monomer. What are oligomerization? I have covered in prior 2 videos, and this is the score, the quality score, and this is the global score and the Z score, and which template was copied. We selected this one. So now if you want to download this model, so download, just here model, you have created one model, so it says 1. Click here and it comes here, and now these are the some other statistics, and this is the alignment, because I told you that based on the homology the template will be selected. Okay, and now if you want to download, so wait, there should be, okay, archive, you can see the download page. You downloaded all in zip files. Now how to open this structure? I had covered 3 videos on DS Viewer, and will also cover more videos on other programs. So right now I can only suggest you those 3 videos to watch on DS Viewer for further analysis of the structure, for to make good figures like that. But that was all how to develop a model for your protein, how to generate model for your protein based on homology modeling. I have also told you how to generate mutation in the structure, just you need to bring changes at the sequence level. So that's it for now. I hope you have enjoyed. If you have any question from this tutorial, please drop it in the comment section, and stay connected for more interesting videos and more interesting tutorials. For now on, bye bye, Allah Hafiz.